I am Professor S. Kripanidhi, Academic Director from Srimaharshi Research Institute of Vedic Technology. I am glad to introduce to you all today's speakers. And SRIVT, on the eve of Silver Jubilee celebrations, is organizing weekly webinars related to Vedic sciences and Indian culture. In this aspect, we are glad to have today with us Professor Reddanna sir, and also along with him, Professor N.B. Brindavanam. So now it is my privilege to introduce both the speakers of today. So Dr. Reddanna, Professor Emeritus in the School of Life Sciences, University of Hyderabad, has over 35 years of research career in the field of inflammation and cancer. He has guided 41 students for PhD and published around 200 papers with a citation more than 10,000 and an H-index of 55. He served as the founder director of National Institute of Animal Biotechnology, popularly called NIAB, and the Dean School of Life Sciences. As the director of Aspire, a Section 8 company promoting innovations and entrepreneurship, he played a key role in promoting innovations, entrepreneurships, and startup ecosystems. He is currently the executive president of the Federation of Asian Biotech Associations, FABA, popularly called FABA, a non-profit organization that promotes academy, industry interactions, career guidance, skill development activities, and startup ecosystems. He was the recipient of Rockefeller's Foundation Biotechnology Career and the Rayon International Foundation Awards. He also received the Outstanding Contribution Award, Pharma, from the Chemtech Foundation and Outstanding Scientist Award for the Benefit of Industry from the Federation of Andhra Pradesh Chamber of Commerce and Industry. So with this brief introduction now, I introduce to you all the second speaker who is getting associated with Professor Reddanna. He is Dr. N.B. Brindavanam. Dr. N.B. Brindavanam is a graduate in Ayurveda and a postgraduate in medicinal plants. And he got the award of PhD in the area of natural resources and sustainable development. He is sharing today's topic, anti-inflammatory profiling of Brihatpanchamoola. Okay, so topic of today along with Professor Reddanna. So Dr. Brindavanam acquired over 3 decades of experience in research and development and took honorable retirement from Dabur in 2017. Dabur is a well-recognized, internationally popular Indian medicine manufacturers. Dr. Brindavanam gained experience in the areas of Ayurvedic pharmaceutical research and bioresource management and regulatory affairs. Particularly, he holds international patents, and again, he's authored several articles and received prestigious awards. So with this brief introduction of both Professor Reddanna and Dr. N.B. Brindavanam, I now request Professor Reddanna to take up the stage. Thank you.
The presidential remarks, by Dr. P.G. Rao Garu.
Yeah, very good afternoon to all of you. Professor Pallu Reddanna Garu and Dr. N.B. Brindavanam Garu. Very warm welcome to you on behalf of SRIVT to this meeting. And it's very touching that SRIVT has completed 25 years of its existence with several difficulties passing through. And Professor Reddanna is very well aware about the growth of the institute, and so also many of the participants who are here. So I would like to have a very outstanding insight lecture from Professor Reddanna and Dr. Brindavanam. And warm welcome to you, sir. We can go ahead with this. Professor Reddanna garu, platform is yours. Please go ahead.
Thank you, Professor P.G. Rao garu. It is an honor, in fact, to speak in this platform. So before I begin, let Dr. Jagadish say a few words about the Federation of Asian Biotech Association, which is partnering with SRIVT in organizing this meeting. Dr. Jagadish.
Good evening, sir. Yeah. Good evening, everybody. Good evening, all the dignitaries. So it's an honor to present here about FABA. So I'll give a few minutes presentation about FABA. Then you can understand what FABA does, and then how you can actually utilize FABA services for anything, maybe conducting webinars like this, or skill development programs that you are aiming at, or even any kind of consulting work. So please allow me to share my screen. Okay, sir, can you see my screen?
Yes.
Yes. Okay, sir. So my name is Dr. Jagadish Ganla. I work as a COO at FABA under Professor Reddanna, sir. So here today, I will tell you what FABA has in store. Like if you know already, FABA is a an organization that is established 20 years back with a mission to basically bridge the gap between industry and academia. Currently, FABA has global presence. We have chapters in 22 countries. We recently also opened a chapter in USA. Even though it's called Federation of Asian Biotech Association, we're happy to say now Asia has been used even in Africa, US, and maybe also in European countries. So we work as we connect various players in pharma and biopharma. As I said, we have many international chapters, and a couple of them are like Bangladesh, Indonesia, Pakistan, Malaysia, Nigeria, Japan, Saudi Arabia, UAE, USA, and many new chapters are also coming up. So FABA is headed by various dignitaries. They span from not only academia, also from industry, startup sector, and also government sector. Currently, we are headed by Dr. Azul Ghani from Bangladesh. And of course, Reddanna sir is our executive president, and we have many other dignitaries, either CCMB director or CDFD director, ICT director. They're all sitting on our board. So if you want to understand what FABA does or how FABA is connected with, FABA is basically connects with everybody under the umbrella of life sciences. So we work with government organizations, incubators, investors, students, startups, institutes, pharma, biopharma companies, university colleges, anybody under the umbrella of life sciences. We have either connections or otherwise, if needed, we will work with them. So just for understanding, FABA has been separated into 3 pillars. Like first pillar is we call it as an ecosystem builder or enabler. I will tell you what exactly it means. So we do various events. For example, during COVID pandemic, we worked with Government of India, also with private companies, how to tackle the supply chain challenges of COVID vaccines, mainly in the Indian imperative. So we worked with government and gave kind of guidelines how the vaccine supply chain has to be handled. That's one of the examples. The another one is we worked with Andhra Pradesh government, mainly to tackle antimicrobial resistance. And WHO has mandated every country to tackle antimicrobial resistance in their own respective ways. And India has mandated that every state should have a state action plan. So Andhra Pradesh government worked with FABA, and then we gave a mandate like how antimicrobial resistance has to be tackled in the whole state. And another one noteworthy event is called Hyderabad Life Science Innovation Cluster Meet, which is a core event that we do where we bring industry and academic people together, mainly all the top-notch companies in Hyderabad and also top-notch institutes in Hyderabad, and then where we allow them to exchange a dialogue where academia and industry can collaborate. So these are kind of couple of events that we do at the ecosystem level. We do many events, but few of them I'm showing here. And another pillar which falls in the category of today's webinar is FABA Academy. In the academic sector, we basically do a lot of workshops, webinars, skill development trainings, hands-on trainings, conferences, job placements, anything that helps either students, faculty, or even industry insiders. We work through FABA Academy. It's all about upskilling people. So we do workshops like drug discovery and development workshop, biologics and biosimilars, chromatography, and many other techniques that are needed in the industry or in academia. Apart from these kind of hard skills, we also work on soft skills like how to make a simple CV, how to apply for jobs, or how to do personal branding. So all these things are not taught in the regular curriculum. That's where FABA steps in and trains people through mentoring sessions. So we had an initiative last year called FABA Fridays. If anybody is interested about like upskilling things, like, how to write a CV, how to write a cover letter, art of networking, and all these things, we have recorded all the sessions and available on our YouTube channel. So anything that is related to personal branding for students or even industry insiders, we have all the skills ready, all the recordings ready, and we have uploaded that on YouTube, and it is available publicly for everybody. So we also have a podcast where we are interviewing experts like you. If anybody is interested in FABA's podcast, please let me know. And the other wing, which is coming up in FABA, is called FABA Entrepreneurship. In FABA Entrepreneurship, we work with startups and we co-create and also help them to raise investments. As you know, in India, many startups, especially in the biotech sector, they may get initial grants from government called Biotechnology Ignition Grant or any other grants. But once the grant is over, they are left alone. Like, there is no venture capital industry that is existing in India for biotech companies. That's where FABA is helping them out to raise big ticket investments. Not only investments, we also provide mentoring, mainly, when there are regulatory affairs, clinical trials, or marketing or anything. We do mainly through our contacts. So if you understand how many people we have impacted, so from past 2 to 3 years, we have impacted over 12,000 people through 100+ events. It could be workshops or webinars. As I said, we have 25+ executive committee board members, and we have 22 international chapters. So upcoming programs, which I think you all can participate, is a drug discovery and development workshop happening in August, from August 3rd to August 16th, followed by Whale Tank 2, where we'll be inviting global venture capitalists and providing them a platform for exposure to Indian startups, which are raising around $1 million+. So we also are planning to do Hyderabad Life Science Innovation Cluster 2 meeting. So for drug discovery workshop, this is the link I'll provide later in our chat. So you can have a look and register to it if you are interested, or otherwise, please send it to somebody who can join the workshop. So in a nutshell, if you want to understand what FABA is a big network having access to industry, academia, and many other stakeholders. With that network, we have an academy to upskill people, to upskill mainly students, thereby bridging the gap between industry and academia. And we have FABA Entrepreneurship helping startups. So how do FABA work? Like, how people can join FABA? So we have a membership model. Like, if we have international chapters, they pay $500 per year to join us. If you're a student, you pay just 1,000 rupees plus GST. Or if you're an individual, if you want to take lifetime membership and work with FABA, so you have 20,000 plus GST to join us. Or if you're an institution, which is 1,77,000, and if you're a startup, it's around 1,18,000. If you're a corporate is the one which we charge more. That's where a lot of our activities, or even FABA's office works through that membership. So thank you. So that is FABA. So FABA in a nutshell is a network. Through academy, we help students, and through entrepreneurship, we help startups.
Yeah, thank you, Dr. Jagdish. I think let me request Divya to share the presentation for me as well as to Dr. Brindavanam. So while Divya shares the presentation, we have been working in the area of anti-inflammatory and anti-cancer drug discovery programs, not only on synthetic products, but also on natural products. As part of this, we have in fact collaborated with Dabur Life Sciences and Dabur Pharma divisions for various anti-inflammatory drug discovery programs. One of that program was mainly on Brihatpanchamoola, where Dr. Brindavanam was the investigator from Dabur, and I was the investigator from University of Hyderabad. And together we have taken a multi-centered investigation on Brihatpanchamoola. And part of my presentation today is the findings on the study that we have taken with the financial support from National Medical Plan Board. With that, I now request Dr. Brindavanam to introduce the Indian traditional system of medicine and also Brihatpanchamoola first. And then later I will take up the rest of the presentation on anti-inflammatory drug discovery.
Thank you very much, Professor Reddanna. Actually, I consider this is a great opportunity to be among the August audience of the institute, Srimaharshi Vedic Research Institute. And I actually thank you for introducing me to this institution per se. For the first time, I think I have an opportunity to interact with luminaries in the field of researching the traditional knowledge systems. I consider that it's a great opportunity for me to be with you today. And I thank the organizers also for including me as a co-speaker to Dr. Reddanna's presentation today. So my scope of work, my scope of presentation today is only limited to drug development and traditional systems of medicine. I will be providing a curtain raiser. And then I would touch upon the Ayurveda and Brihatpanchamoola, which actually set the background for the project. Next slide, please. It is very well understood. It does not require much of an emphasis. The botanical resources have been the healthcare resources and medicines and providing the healthcare services to the primitive to the people and societies in the primitive ages. And since as the human societies evolved intellectually, two distinct kind of medicinal practices emerged into two streams. One is known as the ethnomedicine, that is prevalent all over the world, including African and European countries. It is based upon purely empirical experiences. People know this particular plant is useful for a particular disease or particular ailment. In the second stream, we have codified systems of medicine, which I abbreviated as CSM in this entire presentation. What they have done is the empirical experiences with the medicinal substances available in the nature. They have enriched them with logical interpretations and also providing a philosophical dimensions. That is where the system of Ayurveda came into play. So the evolution of codified systems of medicine is largely witnessed in the eastern parts of the world, particularly India, China, and some of the Middle East countries, though. So in case of the present approach, the modern drugs are basically they comprise of single molecular entities, meaning the drug substance contains only one chemical entity as a single molecular entity, and that entity is recognized as an allopathic medicine, though they don't agree that labeling. It is known as modern system of medicine only. In the modern system of medicine, the tradition is to use the drugs as a single molecular entity. Next slide, please. So, we have many examples. Most of the drug substances that we are in use today are traceable to a plant or a botanical resource, be it aspirin, digoxin, morphine. These were the earliest drugs isolated from the plant kingdom. And the drug discovery from the plants can happen through two different mechanisms. Either you go for a random screening, it's a blind screening, you don't know what is there, but keep on investigating. And then the second is the second way of obtaining taking evidential leads from the ethnomedicine or the codified systems of medicine. I will show you a few examples. Next slide, please. The drugs developed on the ethnomedicinal leads are primarily the quinine, the first one of the earliest drugs isolated from the bark of for malaria treatment, and secondly the vincristine and vinblastine, which are used for today. They are being used for the purpose for treating the cancers, the liquid cancers. Essentially, they were obtained from the Vinca rosea. It was an incidental discovery that actually Vinca rosea was being studied based upon a tribal lead for the treatment of diabetes. In the course of investigation, they observed that actually these two molecules have greater potential as anti-cancer agents, and therefore the course of investigation was changed. In the second category, the Rauwolfia serpentina is very well known case. This is a drug isolated and developed in India based on the Unani systems of medicine. Dr. Ajmal Khan Hakim Ajmal Khan provided this lead for development of reserpine. And then podophyllin is based upon a homeopathic system of medicine. And then artemisinin comes from the Chinese systems of medicine. Statins, which are used for the treatment of cholesterol, lipid disorders, is also derived from the traditional Chinese system. And indeed, the Ayurveda or the Indian systems of medicine also provided lead for the development of vaccine derivatives, which are used in the bronchial asthma today. Now we have the random drug screening is another model by which you can isolate and purify and develop single molecular entities for use as the drugs. The Taxus brevifolia, camptothecin, the anti-cancer drugs coming from these two species were largely based on the random screening. There was no any specific lead that was provided by the codified system or traditional system. So today's scenario is like that. Almost 40% of the drug molecules which are in use today, they are largely derived from the you taking the leads from the traditional medical knowledge. Next slide. Next slide, please. So, and sometimes the plants also serve as a source of drug intermediates. Not every time we go for isolation of the chemical entity the singular molecular entity. Sometimes the precursors are identified, and from precursor we go for semi-synthetic kind of channels. So, for example, the 10-diacetyl becatin. Once upon a time, paclitaxel was directly isolated from Taxus. However, we found that the paclitaxel isolation is costly affair. So, therefore, to improve the economy, we went back to the first one precursor, the 10-diacetyl becatin. And once if we are able to synthesize paclitaxel, semi-synthetic route from becatin, it also gives an opportunity to isolate another, to develop another semi-synthetic analog, docetaxel. Like that, sometimes I need not get the final molecule, but sometimes the precursors will also help us to obtain the molecule. Therefore, that is the reason most of the modern molecules are dependent on the plant kingdom at one point or the other time. Next slide. Next slide, please. So now the question comes, I have illustrated number of cases related to the leads provided by the traditional systems of medicine like from China or Unani. But what actually, whether we were able to provide any kind of meaningful leads for drug development from Ayurveda, which is the Indian systems of medicine, we have fairly good number of molecules to illustrate. One is the vaccine I have already mentioned. Now, colchicine is another molecule, Gloriosa superba. It is an indigenous Indian medicinal plant, which is being cultivated extensively for production of colchicine. And another molecule that was discovered for BP treatment was based upon an Ayurvedic medicinal plant known as Makandi. And today we have large number of phytopharmaceutical products. In the phytopharmaceutical products, they are not actually the single molecular entities. They contain the single molecular entity is captured and in a matrix of other chemical substances. This particular kind of a novel set of products have wider applications. They can be used as nutraceuticals, cosmeceuticals, and different. So Ayurveda has provided large number of leads for this group of products, and they are highly prevalent and they are highly demanded products in the global markets. The bacosides rich Brahmi extract or the HCA hydroxycitric acid rich Garcinia extract, boswellic acid, curcumin, the haridra, the turmeric is now a most popular nutraceutical product or cosmeceutical product available in the global markets. The leads were obtained from based on the Ayurvedic traditional system. Next slide. Having said that, it is also important that we consider the drug development as an avenue for the enrichment of Ayush systems of medicine and how they would benefit the country on a long run. The codified systems of medicine in India have been pluralistic. I do not want to get into the historical reason. Ayurveda was the original systems of medicine that was born and evolved in India. But later, country was accommodative to welcome the Unani systems of medicine. due to various political, historical and political reasons. Similarly, homeopathy systems of medicine entered, and also today the country is welcoming the products and formulations based on the traditional Chinese TCM, traditional Chinese systems of medicine. And to conglomerate all these effects to bring synergies among all these different systems of codified systems of medicine, Government of India has constituted an independent Ministry of Ayush for multifaceted development. Ayush covers Ayurveda, Unani, Yoga, Siddha, and homeopathy. And there is a great global enthusiasm about the Ayush streams of the medicine because they are seen as a potential solution to the emerging healthcare problems in the global healthcare scenario. And we also see multiple opportunities for development of new drugs, nutraceuticals, and healthcare supplements using modern scientific tools. This is where the focus of Government of India is now concentrated upon. Next one. Currently, the global and national initiatives, we have two core objectives to bring back the glory for the Ayurveda and Indian systems of medicine with a global acceptance. Today, mostly we are utilizing the traditional knowledge systems much in India, but its global acceptance yet to be upgraded and upscaled further. And we need, for that purpose, we need to fill some prevailing scientific need gaps so that we are able to overcome the regulatory barriers and technological barriers. So there are a number of initiatives which we have listed. There is a network of projects for which Dr. Reddanna has been a very major partner. The CSR network project, the Council of Scientific and Industrial Research. And there are 25, the country has signed up 25 country-to-country memorandum of understanding for cooperation in the field of traditional medicine. And Indo-US initiative, this has been a very great initiative for the discovery of very specifically the cancer, anti-cancer drugs based upon the Ayush systems of medicine. And it is not just about the anti-cancer molecules. It also talks about the adjuvant therapies to be given in the cancer treatment who are undergoing chemotherapy or cancer. Next one. So the global summit, recently the World Health Organization also hosted a global summit in India. Now, given that kind of, this sets the background as to the importance of natural products and botanical resources for drug development. And the future of the drugs will be largely dependent on the health of our biological diversity. Next, I touch upon the second aspect of my talk today is the Ayurveda and Brihatpanchamoola part of it. Next slide, please. Having said about the Ayurveda, I think I didn't introduce the system of medicine. I also feel that it is really does not require much of a very elaborate introduction to an institution which works on the traditional knowledge of systems. Like many of our Vedic knowledge systems, origin of Ayurveda is linked to the divine forces. So therefore, we cannot say exactly what was where the origins of the Ayurveda are not traceable that way. But the first document of this codified system, Charaka Samhita, is estimated to be something like 3,500 years old. Something like 1500 BC, the first treatise on the Ayurvedic systems of medicine was published by Indian sages. The entire Ayurvedic systems of medicine, what actually codified systems of medicine, whether it is Chinese or the Ayurveda or Unani, they don't follow the same kind of doctrines which are used in the modern systems of the medicine. They have their own historicity, they have their own epistemology, and they have their own kind of basic doctrines on which the entire systems of entire knowledge system has been structured and built up. In the Ayurveda, similarly, we believe actually the entire core doctrine of Ayurveda is well reflected in the Sankhya philosophy. Most of our Indian traditional knowledge systems do agree that entire universe, the entire matter existing on the planet is derived from five primordial elements known as the Panchamahabhutas. And they, because of the configuration and because of the ratios that each of these bhutas represent, the matter becomes diversified and different. So therefore, the Ayurveda also believed that the primordial elements constitute the human body, and the same primordial elements constitute the rest of the matter in the universe. So therefore, there is a structural and functional interrelationship between the human body and rest of it. So exactly what Ayurveda does is it goes deep into the healthcare applications of rest of various substances, be it plants or minerals or even metals. How they can be applied to the human health is on the basis of the structural and functional interrelationships, which are created by the ratios and the configurations of the 5 primordial elements. And I am sure the members of this audience would realize, because I was given to understand you have been working on the metallurgy part of it, and in the metallurgy also the concept of this 5 primordial elements plays a very, very important role. Here the only difference is we study the primary human body with reference to primordial manifestations, and we study the manifestations of the rest of the universe, and we try to marry these manifestations in the favor of healthcare. Next slide. Next slide, please. So, more interestingly, though it is very old ancient systems of medicine, almost aged to 4,000 years of traceable history, the hallmark of Ayurveda is this pharmaceutical process. It is not something like that you pluck a plant and administer, or take a fresh juice out of that and administer to treat a particular disease. The system is equipped with sophisticated pharmaceutical processes, and we have very well evolved drug delivery systems defined in the textbooks of the medieval periods. And incidentally, both the drug and drug delivery system, they are inseparable. Sometime in future, I would like to certainly deal with these aspects more in detail, but today I would like to confine with these simple statements only. More interestingly, if we talk to the phytochemist or if we talk to a drug developer, he recognizes that each individual herb is a polypharmacy because it contains more than one single molecule which is biologically active. And further, the Ayurveda complicates the system further by opting for a concept called the polyherbalism in its pharmaceutical practices. Here, what we try to do is, instead of using one single herb, we take multiple herbs to formulate one. And one particular herb goes into multiple formulations also. It divides both the scenario. So the therapeutic applications of individual formulations vary very greatly, and they exhibit diverse pharmacological activities. And in addition to that, the Ayurveda also made a unique proposition in terms of the pharmaceutical practices is the fixed dose combinations. In the fixed dose combination, we have 3 to 10 ingredients like Triphala, or we call Dashamoola. I can use Triphala as one group, or I can use Triphala as a subgroup in some other product also. So this is the level of complexity we have created for promotion of the polyherbal formulations, and they have tremendous advantages both from the human health point of view and also from the ecological management point of view. Next slide, please. Now I come to the background of this particular study, which Dr. Reddanna would be elaborating further. This particular starting point, we took a lead from the concept of Abhava Pratinidhi. What happened is, in my introduction, it was told that I was required to manage the biological resources or the medicinal plants in a sustainable manner. And that is where I actually looked into the concept of Abhava Pratinidhi and its importance in the sustainable management of the medicinal plant resources. So the concept actually provides a procedure to replace or substitute a plant substance or a plant part which is unsustainable or which is not available. If you could easily imagine 4,000 years ago, the system has prescribed something, and today the plants described in that period may or may not be available, and we must have lost some of them. So therefore, it provided an opportunity to identify substitutions for the lost or extinct or the endangered medicinal plants. And the basis is, this recommendation is very simple. You should draw very specific structural and functional correlations between the new and the old substance. And there are multiple, if we look at the Ayurvedic textbooks, we could read as good as 125 examples of mutual substitutions. So the scientific, we try to understand the concept very scientifically, and number of investigations were carried in this direction. Very interestingly, Aconitum heterophyllum and Cyperus scariosus, we found excellent kind of biological activity correlations. Similarly, in Emilia ribes and Emilia xerium cotum, we found that the chemotaxonomical correlations between the. So this is the beginning point for me. Why we went to Brihatpanchamoola project. In next slide, I would close the discussion from my part. And this particular approach also give an advantage for new drug development. But today, for example, the embelin is being evaluated again as a therapeutic option in the weight management. Next slide, please. So now the study candidate Brihatpanchamoola is based upon this particular Abhava Pratinidhi concept. I find that there is a tremendous sustainability risk in the group. Dashamoola is a fixed dose combination. It contains 10 medicinal plants actually. And out of 10, there are five species are the trees, are the shrubs by their habit. And rest of them are the 5 herbaceous species. So Brihatpanchamoola refers to 5 root drugs of the tree species. Brihat is the bigger, Pancha is the 5, and Moola is the root. So the bigger 5 roots, and the Laghu Panchamoola, the smaller 5 roots. This is the combination we take as a Dashamoola. Incidentally, Brihatpanchamoola is the individual ingredients are not used in there as a standalone entities. There are at least 4 species covered in the Brihatpanchamoola. 2 of the tree species and 2 of the herbaceous species were never used as a standalone ingredients. They are always used in the combination, in this fixed dose combination only. That shows actually scientifically they must be associated with some degree of untoward effects or insufficiency in the functional outcomes by their usage. Therefore, they are combined with other ingredients. Perhaps that could be the logic. So this is, but today's bigger problem is availability of the roots of the bigger trees for therapeutic applications on large volumes. The market demands for the 5 Brihatpanchamoola candidates is something like 5,000 metric tons of dry root bark or stem bark in the on a long run. So on a continuous basis. Next slide. So that is why we felt at Dabur, we felt that this Brihatpanchamoola could be the best candidate for investigation to identify Abhava Pratinidhi drugs. And we thought actually our hypothesis was, instead of using a stem bark, if I am able to use root bark, younger roots, where I don't have to destroy a grown-up tree. Idea is that we multiply the plants in a high density storm condition, harvest the younger roots, and whether these younger roots can be used as a substitute for the. So therefore, the present study was aimed to evaluate the younger roots as an alternative plant bark. In a Sanskrit terminology, younger roots is the Abhava Pratinidhi Dravya for the root or stem bark to be used in the Ayurvedic medicines. So therefore, we chosen the biological activity based, and Professor Reddanna used a wide battery of anti-inflammatory models, molecular models for describing the protocols. I wish, I think I have already taken much of a time in partnering with Dr. Reddanna. Now I request Dr. Reddanna to describe the entire study protocol.
Thank you very much, Dr. Brindavanam, for laying the foundation and also introducing the traditional systems of medicine and also the Brihatpanchamoola. So, as you highlighted, there is enormous demand for these plant species, specifically for various Ayurvedic formulation development. And as you said, the sustainability of these raw materials is the major problem, constraint. And any replacement, it has to be on a scientific basis. If at all any of these plant species which are not available and sustainable sources have to be provided, then it has to be on a rational basis. With that basis in mind only, in fact, the proposal that we both submitted to the National Medicinal Plant Board is mainly to come up with alternative sources of the raw materials for the Brihatpanchamoola, which are being used widely for anti-inflammatory formulations. Next. So inflammation is the major component in many of the disorders. Whatever may be the type of disease that you are talking, inflammation is a component. It is a cellular, immune, and metabolic response to injury or infection. It is usually a protective vascular connective tissue reaction, mainly aimed to eliminate the cause of injury and clean up the dead and dying cells. But at the same time, the next slide, please. So, as long as this inflammation is under containment, there is no problem. But the chronic inflammation, when it goes beyond the control, inflammation leads to a variety of disorders. Whatever may be the disorder that we are talking, inflammation is involved. And in this inflammatory process, the major players which are involved in mediating the inflammation are some of the bioactive lipids, which are called as eicosanoids, which are derived from 20 carbon chain length fatty acids. And prostaglandins and other related molecules have been termed as eicosanoids. They are all derived from a 20 carbon chain length fatty acid, and they act as local hormones. They are produced at the site where the inflammation is taking place, and they are eliminated within few minutes, sometimes even seconds, after the action is done. But in addition to, but these local hormones are, as I said, they are locally produced to elicit the response against whatever the infection or injury that has taken place. So in addition to participating in various intracellular signaling, they're also involved in intracellular signaling cascades also. And the major players in this pathway are the prostaglandins and its related. So to explain about the formation, I'm sure everybody is aware of the cell membrane, which is formed from phospholipid molecules. And this phospholipid molecule contains one unsaturated fatty acid, and one of that is the arachidonic acid, which I was mentioning, 20 carbon chain length fatty acid. And in response to injury or infection, the fatty acid is released from the membrane phospholipids by the action of phospholipases. And once the arachidonic acid is released from the membrane phospholipids, and it is acted upon by a series of enzymes, one such enzyme is the cyclooxygenase, which incorporates two molecules of oxygen and then converts into a prostaglandin H2 molecule. And this prostaglandin H2 forms the precursor for variety of tissues to produce variety of the molecules. For inflammation, the key player is the prostaglandin E2, which is formed from the prostaglandin H2 in all inflammatory conditions. And that is the one which mediates the pain, fever, redness, swelling that we see, all the inflammatory symptoms. Next. And similarly, in addition to this, there is another pathway which we are so in response to such kind of infection or injury, the phospholipases releases the fatty acid. But here there is another pathway which is called as lipoxygenase pathway, which incorporates one molecule of oxygen and produces the leukotrienes, and these are the major mediators of allergy asthma. While prostaglandin E2 is the major mediator of inflammation, leukotrienes are the major mediator of allergy and asthma. And whatever the non-steroidal anti-inflammatory drugs that we are taking today, they inhibit the prostaglandin E2 formation. And the anti-asthmatic drugs that we take today, the montelukast, gefidlukast, Montex, anybody who goes to the doctor with respiratory problems, they will be put on Montex, and that is nothing but the leukotriene receptor antagonist. And in fact, there is one natural product which has come up, which is a lipoxygenase inhibitor, gelatin, is a plant-derived product which inhibits the lipoxygenase, and it was there in the market also. Similarly, with the prostaglandins also, aspirin, which is acetyl salicylic acid, inhibits the prostaglandin formation. That is the one of the first, first molecule anti-inflammatory drug that came into the market, derived from the plant species. And even today, aspirin is the most popular drug being used for a variety of applications, not only in inflammation, but as blood thinner and many other in case of the cardiovascular problems. Next. So, as I was mentioning, a number of these eicosanoids, which are nothing but the 20-carbon chain length fatty acid derived oxygenated metabolites, they play in each and every role of inflammation, either vasoconstriction, vasodilation, increased vascular permeability, chemotaxis, bronchospasm, platelet aggregation, pain mediation, fever induction. These are all the symptoms that you see in inflammatory condition. And these eicosanoids or the bioactive lipids are the ones which are playing a role. And as a result, these anti-inflammatory drugs have their origin from eicosanoids. Next. So, as I was mentioning, the good side is inflammation is designed to eliminate the cause of injury or infection, to clean up the dead and dying cells. That is the good side of inflammation. But the bad side is often this inflammatory response is out of proportion to the threat it is dealing with. And the result can be more damaging than the original cause of the injury itself. And so that's the kind of it is, inflammation is like a fire. As long as it is under control, it could be used for a number of beneficial purposes. But once it goes out of control, the fire, it can devastate, damage everything that comes across. Similarly, inflammation is also a process causing such kind of damage when it goes out of control. Next. So I was mentioning earlier, in every type of disease, inflammation has a role, like Alzheimer's, anaphylaxis, asthma, atherosclerosis, atopic dermatitis, including various cardiovascular disorders and cancer. In all of them, inflammation has a role. Whatever may be the root cause for initiation of the disease, but the progression at least is associated with the inflammatory process. And that is where the anti-inflammatory drugs have become very important, and they are the most predominant medications that you see in the healthcare sector. Next. So inflammation also has a role in cancer. Specifically, the chronic inflammation is the one which leads to, if it is not addressed, it may lead to a variety of cancers. Next. And inflammation, as a result of this, inflammation has been introduced as the seventh hallmark of cancer. In any cancer tissue, there is a cancer associated inflammation is there, and specifically the inflammatory environment that is prevailing around the cancer cell is the one which is causing much more damage than the cancer itself, because it helps in the metastasis of the spreading of the cancer tissues to other tissues also. So that is where inflammation has been introduced as the seventh hallmark of cancer. Next. So as I was mentioning, in all the cyclooxygenase 2 is the one which is involved in mediating inflammation or formation of the prostaglandins. And this COX2 was implicated in variety of cancers. It is involved in overexpression of the COX2 in variety of cancers. And as a result, today, anti-inflammatory drugs are also being used in the treatment of some of the cancers, specifically colon cancer. Next. So, as the normal epithelium, when the normal cell is getting converted into a neoplastic cell, during its transformation from early to late stages, you will see the COX2 expression goes up. And that is associated with metastasis and other kind of proliferation that you see, along with other oncogenes that you see in a typical cancer tissue. So not only in normal inflammatory disorders, but also in cancer, inflammation plays a role. Next. So with that, as in the light of this, there is enormous demand for the anti-inflammatory drugs. And so not only in the allopathic system of medicine, but also in the traditional system of medicine. As Dr. Brindavanam was explaining, a number of these Brihatpanchamoola, the big root bark or stem barks, are being used in various anti-inflammatory formulations in Ayurvedic system of medicine. And as a result, they are being overexploited, and there is a depth of the demand for this stem bark or root bark has resulted in endangering these species, and the availability of raw material has become a major problem. In the light of that, National Medicine Plant Board also has come up, initiated a program to come up with alternatives to stem bark and root bark. So in that connection only, so myself and from Dr. Brindavanam from Dauber, we together have submitted a major proposal, and then also involved other institutions into this program, and that has resulted in a multicentric investigation on Brihatpanchamoola, which I wanted to present today. Next. That is the multicentric investigations on Brihatpanchamoola with special reference to anti-inflammatory profiling of the young roots. Next. So to compare the anti-inflammatory activities of young roots and leaf extracts with stem and root bark, which is originally being prescribed, the various from these different plant species. So the objective was to compare the anti-inflammatory activities of the traditionally used materials like stem bark and root bark with alternatives that are being proposed, that is young roots and leaf extracts were taken. These are the various species. Next. The study was designed in such a way that various extracts of different parts of the 8 plant species collected from different places, not only at one place like Gujarat, Chandigarh, Orissa, Andhra Pradesh, Maharashtra, Madhya Pradesh, Karnataka, were provided by Dauber. So Dr. Brindavanam played a key role not only in raising these plants in different states, but also harvesting them and coming up with the extracts, which are the same way that they are going to use in Ayurvedic formulations, and then they were supplied to us. At University of Hyderabad, our team was mainly involved in vitro anti-inflammatory potentials of these extracts were evaluated. How we have done that? A battery of various enzymatic pathways and other cytokines and chemokines were evaluated. So 5-lipoxygenase, which I was mentioning, is the one which is the major mediator of allergy and asthma. Cyclooxygenase, COX-2, COX-1, they are the targets for inflammation. They are involved in prostaglandin biosynthesis. So we have evaluated the inhibitory potential of these extracts of these enzymes. Then macrophage cytotoxicity assays. Macrophages are the key players in inflammation, and we have evaluated the effects of these extracts on the macrophages in the cells that were grown in the laboratory. And we also evaluated induction of specific immunity by lymphocyte proliferation assay. Lymphocyte proliferation assay is the one which is mainly used for evaluating the immune response of the extracts. Either they are boosting the immunity or suppressing the immunity, things like that. Pro-inflammatory cytokines like macrophage infiltration, the protein alpha, MIP, interleukins, and prostaglandin E2, all these were evaluated. And anti-inflammatory cytokines, IL-2 also, induction, pro and anti-inflammatory cytokines were evaluated in such kind of in vitro assay systems. Next. As I was mentioning, these extracts, these plants were grown in different parts of the country under natural conditions. Next. And the plant parts that we have used in this study is root bark and stem bark, which are traditionally used. And then to replace them, we have evaluated leaves and the young roots. Young roots again, half a year, 1 year, 1.5 year, 2 years, and 3 years, different time. And what kind of extracts? Petroleum ether, ethyl acetate, ethanol, and aqueous. It became a humongous effort in coming up with various uh, extracts, and then also for each extract, again, different solvent systems we have used. Next. So inhibitory assays and oxygenases, enzyme inhibitory assays, 5-lipoxygenase, cyclooxygenase 1, and those in all this, and this is the major target for anti-inflammatory drugs development. In fact, a number of COX-2 specific drugs are in the market, like celecoxib, roficoxib, and a number of coxibs are there. And any extract which is showing inhibitory potential of the COX-2, then we have evaluated cytokine analysis, the prostaglandin E2 and IL-1 beta, IL-6 and MIP-1 alpha and IL-2. All these pro and anti-inflammatory mediators were analyzed. Next. And almost 604 extracts we have evaluated for various biochemical parameters. And in this, 224 extracts were screened for COX-2 inhibition, COX-LOX dual inhibitors, and mitogenic activity and lymphocyte proliferation assay. So we have identified which are the potential extracts for COX-2 inhibition, and also COX and LOX dual inhibition, and also which are involved in lymphocyte proliferation assay. Next. So final conclusions drawn from comparing the leaves and the roots of young plants with those of the adult stem and root barks are, so in case of Sinoca bean, ethyl acetate and ethanol extracts of the leaves, petroleum ether and ethyl acetate extracts of the young roots um, can become alternative sources for anti-inflammatory act. So for every plant, we have identified what could be the best component for the replacement of the adult stem or root barks. And also, if they are not replacing, whether they are 50% efficacy, or sometimes they may be they are much more potent than the adult stem or root bark extracts. So we have evaluated like that, and then compiled all these studies, and then a recommendation was submitted to the National Medicinal Plant Board for implementation across the country. Next. So similarly, with reference to Padala, then Agnimantha Lagu, Bilwa, Sayanok, the results were based on these experimental studies. We have sent specific recommendations to them. Next. So from all these studies, we have concluded that anti-inflammatory properties exhibited by specific extracts of young roots are very much comparable to the mature plant pots in most of the cases, but not in all. And also, where in other cases, it is possible to titrate the dosages in young roots to match the levels that we see in adult roots. Suppose if they are only 50% efficacious, then the dose could be escalated to match the level that you see in adult roots. So that is another component. And with the data on COX-2 and 5-LOX dual inhibition by the specific fractions, it is possible to develop formulations with the least side effects. And many of you may remember that COX-2 inhibitors that came into the market, specifically celecoxib, ruficoxib, and other coxibs, when they were given to arthritis patients who have to use for many years, on long-term use of these coxibs, cardiac side effects were seen in some of the patients. And as a result, in fact, one of the coxib, that is Vioxx, was withdrawn from the market. And so the next generation of anti-inflammatory drugs, they have been focusing on COX-2, 5-LOX dual inhibition. And in that connection, we got a very important lead that some of the extracts have shown both COX-2 and 5-LOX dual inhibition, and which could form potential anti-inflammatory components without side effects. That was another major development that we have seen. As maintaining of young saplings is easy and sustainable compared to the mature plants, it is possible to achieve sustainable supply of raw materials for Ayurvedic formulation through a systematic scientific evaluation for their safety and efficacy. So these studies on anti-inflammatory profiling of Brihatpanchamoola have given us several leads for target-based drug development, specifically with reference to anti-inflammatory drugs. So it is not only just we have submitted the report to the National Medicinal Plant Board, but as part of this study, we got several leads which we have taken up for coming up with a single molecule or a group of molecules for developing several kind of anti-inflammatory or anti-cancer drug candidates. So that is with reference to specifically the study that we have taken up with the Dauber on Brihatpanchamoola. Next. And our lab has been working on natural products, a number of them. In fact, out of this, we have come up in some of the cases a single molecule inhibiting the particular target, which could be taken up for further development as per allopathic norms, like specifically with reference to Ficus indicus, a betanin, a molecule was found to be a potent inhibitor of 5-lipoxygenase. And gallic acid is a major component in many of the plant species, specifically Terminalia chebula, Terminalia bellerica. In that, this gallic acid is a potent inhibitor of COX-2. And in fact, we have come up with derivatives of the gallic acid. And because of the hydroxyl groups that are there, we can derive the several derivatives were made, methyl, ethyl, propyl, like that. And those molecules were, in fact, evaluated for COX-LOX dual inhibition, which could form a potential anti-inflammatory without side effects. Similarly, Acacia farnesium, and in some of the cases, we could not come up with a single molecule. But even though the extracts are showing the efficacy, but we could not come up with in case of the Premna integrifolia, we came up with a single molecule. And also in chebulagic acid, in Terminalia chebula, we have come up with a single molecule, identified. And another important major work that we have taken up is on spirulina, the blue-green algae. The blue-green algae contains a copper-containing protein, which is called as phycocyanin. And this phycocyanin is potent. In fact, our group is the first one which shown that it is a potent inhibitor of cyclooxygenase 2, showing anti-inflammatory properties. And also, a number of studies were conducted. In the next slide. Yeah, this is about the study that I was mentioning on Openshia fruits. They have a very good, and we could isolate a potent molecule from this Openshia fruits, which is betanin, inhibiting the 5-lipoxygenase, which could form a potential candidate for asthma allergy. Next. And chebulagic acid from Terminalia chebula, which I was mentioning, it is, in fact, from this we have identified, the next slide, we have identified a COX-LOX dual inhibitor, which could form a better anti-inflammatory drug candidate than we see in the market today. Next. And similarly, isolation and characterization of another COX-LOX dual inhibitor from the Premna integrifolia, Agnimantha Brihat, one of the Brihatpanchamoola candidates. Next. And from this, we have isolated 6-hydroxysalvinolone. So these are all the leads that we got from the study that we have taken up as part of the National Medicinal Plant Board project on Brihatpanchamoola. So all the way going up and identifying a single molecule which could form a potential molecule candidate for drug development. Next. These are the studies on phycocyanin, which I was mentioning, the spirulina platensis. Next. So a series of studies were conducted, and we have published a number of papers on this and shown efficacy in terms of anti-inflammatory and also anti-cancer activities. In fact, in one of the studies we have shown that it can cross the blood-brain barrier and can also take up what is called as the thioacetamide-induced hepatic encephalopathy. When you give thioacetamide to the rats, within a week they die. But along with that, with this, the phycocyanin is given. In fact, it the mice survived for many months. The rats survived. So it is a very good protective candidate for hepatic encephalopathy. Whenever the liver gets damaged, ammonia gets accumulated, and because of the ammonia, the brain gets the toxicity, and they will go into the coma kind of condition. But this protects so well. C. phycocyanin we have shown in animal models. Next. So, in fact, one of the anti-allergy, anti-asthmatic candidate in collaboration with the Ayurvedic company based in Varanasi, we have come up. And in fact, this product, is a Broncho Tea, is a powder that is available in the market, and it could be taken like any other tea powder. Replace that with Broncho Tea, and then you can add milk and sugar, and then you take it up. Broncho tea is available, and it is one of the very good candidates specifically in respiratory diseases during cold conditions. In the north specifically, it is very well, it is available in Amazon and also through Surya Pharmaceutical Company. Next. So that's, next. In conclusion, what natural products and their structural analogs have historically made a major contribution to pharmacotherapy, especially for cancer and other infectious diseases, including inflammatory disorder. Natural products, however, present challenges for drug discovery, such as technical barriers to screening, isolation, characterization, and optimization. Next. And in recent years, several technological and scientific developments in analytical tools like genome mining, engineering strategies, microbial culturing advances are addressing such challenges and opened up new avenues and opportunities. Consequently, it is interest in natural products as drug leads is being revitalized. There is growing interest. And so many of the allopathic drugs are having the side effects as it was experienced. And so any such kind of leads that are available from the traditional systems of medicine, if they are scientifically validated in terms of their efficacy and safety, even if they are not having a single molecule, there will be a global acceptance. And then I think that these traditional systems of medicine will be able to address many of the challenges which allopathic systems are not able to address the challenges. Next. So I think at this point, So allopathic drugs, specifically if we take into cancer into consideration, all cancer or infectious diseases, always the focus has been to kill the cancer cell or to kill the pathogen that is involved. And in this process, not only you are killing the pathogen, but also you are killing the normal healthy cells also. The same happens in chemotherapy when you are using them for treatment of cancers. And that is why unbearable side effects are seen for the cancer patients undergoing chemotherapy. So the focus has been now shifted, rather than killing the cells, empower the body, boost the immune system. And if we boost the immune system, our immune cells will be able to go and kill the cancer cells or kill the invading pathogen. So that has become the emphasis, that has become the focus even in the allopathy system. And that is where immunological therapies have come into picture. And in that connection, the traditional system of medicine offers a number of leads which will boost the immune system. But only we need to evaluate and then show efficacy and safety. Then there will be a global acceptance, and there will be much more wider acceptance to these kind of traditional systems of medicine. With that, I stop, and I will be happy to answer if there are any questions. Along with me, Dr. Brindavanam is also there. Will be very happy to address.
Professor Reddanna garu and Professor Brindavanam. First of all, my appreciation to both of you for taking us through a valuable information and the work that you are doing. My main appreciation is not for the work that you have presented here, but the very thought that you both of you coming together, one from the medicinal plant area and the other from the chemistry and the other aspects of developing drug development, coming together. That's a very great thing that has made most of the time the success depends on such collaborative works only. When we work in isolation, many of the results we were not getting, and that's where the good thought, you have taken this project and you are very successful. And also I appreciate that the very reason that the SRIVT, Maharshi Research Institute of Vedic Technology, works on bringing the traditional knowledge and the modern science together. And you have fulfilled that objective very much knowingly or unknowingly. But the work that you are doing is going to give results a long way. For that, my appreciation, and also thank to FABA for their support in this. And now I request Professor Dr. Kupanidhi sir to take over to take the questions, moderate the question session from now. Thank you very much.
Thank you, Rao Garu. So when Dr. Sastry approached me to give a talk, then I realized to relate my talk to the Vedic Institute that Dr. Sastry has been associated, and he was the founder director, and I thought that I should involve Dr. Brindavanam into this. And Dr. Sastry immediately agreed. And I must thank Sastry for providing this platform, and also Dr. Brindavanam coming forward, joining me in this.
Thank you so much, Professor Reddanna garu and Dr. Brindavanam garu. Actually, this institute, since its inception, 1999, our main theme is bridging gap between Sanatana and Sanootana. Sanatana is ancient sciences and Sanootana is modern sciences. So last 25 years, we are working on the bridging gap between these two fields of the scientific branches, scientific fields. Actually, we also in the natural products, Ayurvedic and scientifically proven Ayurvedic, and we also worked on so many 400 nearly herbal particles. And we also developed two drugs evaluated by NIPER Gauhati. Dr. P.G. Rao Garu helped us to make it done. And we also applied for patent also. So I am very happy that this is the platform. They like this. I wish always to come together. Scholar, Vedic, now this is ancient sciences, scholars, and modern scientists together. We have to work together. Then only things will come out, because so many things to be explored. I believe in future, so many interactions, collaborations, and joint initiatives can be started with this. And I also request Brindavanam ji also extend your support to take it forward. Now, I request Dr. Krupanidhi Garu to take the question session.
Thank you, Director, sir. So many thanks to Professor Reddanna and Professor Brindavanam Garu for having given wonderful presentations on bridging the gap between, as indicated by the Director, Sanatana and Sanootana. So the traditional system of knowledge is in practice, was in practice, and ever been practiced in India. And the impact of this traditional system of practice and the, what is it called, the modern molecular biology and genetic engineering aspects were well elucidated by Professor Reddanna Garu. So now it suits very well the bridging of ancient system to the modern system. Now the what is it called? The presentation is open for discussion, and I request all the viewers to put up whichever the question you feel like. It is open for discussion now.
Questions may be brief, in brief, so that we have time for discussion.
Professor Reddanna Garu, and again our Brindavanam, you both were mentioning about the conservation of the biodiversity, and there is a lot of demand. There is tons of the plants have been required to prepare the medicines. And in this line also we are given a clue that young saplings to some extent, what is it called, suits the dose. So now the medicinal companies, Ayurvedic medicinal companies, are they satisfying with the young saplings? Are they really advocating the government to have the conservation of the biodiversity in a large scale?
I think Dr. Brindavanam.
Yeah, thank you for that question. Actually, the concept was that we are able to produce it is possible, technically possible, to produce the younger roots in a high density short-term plantations. Already work is happening, something like 4 to 5 tons a year into 5 species.
Tons a year, young plants.
Young plants we are producing and harvesting and provided. And I'm very happy to inform you, Dabur as a company has replaced the use of stem and root bars into 5 classical Ayurvedic products like Dashamoola, Thailam kind of. They were actually Dashamoola is a prime ingredient. Presently, they are confining this approach to the medicines which are used for the topical applications and local uses. They have not gone into the products meant for the oral indications as yet. they are also actually the two things we need to address concurrently. One, that we need to increase the scale up the volume of production at the ground level, and also do a little more study on the product containing the younger roots, by very particularly for the long-term toxicity. That is one thing which we need to establish before it is actually introduced into two major products they are contemplating, the Dashamoola Rishta and Chyawanprash. Both these things, we have to, the volume uptakes into these two products are very large. Presently, our production capacity is only 5 tons each into 5 species, 25 tons. So we are contemplating something like 150 tons cumulatively, 30 tons into 5 species.
Sir, is there curation for this germplasm of this endangered plant, sir?
Actually, the biodiversity management for the Ayurvedic medicine plant itself is a very big area of discussion and actions. Presently, National Medicinal Plant Board is having certain kind of schemes. For example, they support the forest departments for the in situ conservation and also the in situ resource augmentation for the important and endangered medicinal plants. Almost under each project, board provides provides a funding of 10 crores to the respective state forestry departments. So under that, they have established fairly very good models. I would say very good models for the in situ conservation of the red listed medicinal trees presently. Okay. So some work is happening. However, we know we need to go a very long way with reference to the resource augmentation. Another important contribution made by the National Medicinal Plant Board has been introducing forest species into the farming conditions. Even at Dabur also, with some marginal financial assistance from National Medicinal Plant Board, we have brought 2 forest species into the purely farming conditions, and all 100% volumes by Dabur are now being sourced through purely cultivation. They stopped harvesting this plant from the forest area. So that's, I would say, beginnings have been made, multiple beginnings have been made in multiple directions, but they need to be scaled up and expanded to cater to the rest of the industry as well.
So farming conditions means this sapling development.
You cultivate them. For example, Uraria picta, we were originally it was native to the lower Gangetic plain forest areas. Now it has disappeared because of multiple pressures. Yes. There is a tremendous biotic stress on the species, plus the harvesting for the commercial volumes. Both these things together, they have made the species highly vulnerable. So we intervened and then started cultivating in the adjoining areas of Kushinagar, Sitapur districts of UP. And the cultivation technique is also highly amenable and economic, highly rewarding to the farmers. So therefore, both the species are now being cultivated. To the extent that Dabur has entered into contractual production arrangement with the farming groups. And the farmers are willing and they are happy to do more, large number of farmers are willing to join the production, provided there is a marketing avenue created for them.
Thank you, sir. Now the questions are solicited from the viewers.
Dr. Brindavanam, is there any idea about to reach this demand and all those things? Can we think about, have you thought about this nanomization of herbal powders? Sir, because when this size is increased in the nano size, the usage will be very low. For example, instead of 1 gram usage, you can take 100 mg. Actually, we have done it for some American company, nanomization of herbal powders, single powders, even polyherbal formulations also. Is it a solution? You think this is a solution?
Yeah, definitely, it is a very viable alternative. However, today we should look at the current regulatory scenario and the licensing requirements. Today we have 2 classes of medicine, Ayurvedic medicines, Ayush products, Ayurveda, Unani, Siddha medicines particularly. One is the classical Ayurvedic medicine. The prescription is that the first category of the medicines known as the classical Ayurvedic medicines, they are based upon the ancient treatises. whatever has been written in the ancient treatises,
As it is, we have to take it.
we have to go by that only. Any kind of alteration would not be permissible, because the purpose of this particular category of products is to keep the spirit of the traditional knowledge system. Else, once if we forget the classical Ayurvedic products, the system of Ayurveda itself goes into oblivion. That is the purpose. Therefore,
You can submit under proprietary category.
but proprietary products, it is always welcome. Any product which is meant to large volume consumption by the consumers should be allowed to go for process innovations and product innovations. And the system is open for that, actually.
Yes. I know that. Right.
So even the Ayush also maybe should open up for such kind of innovations also. A proposal could be submitted to Ayush for the acceptance of such kind of nano herbal formulations also. And
Actually, we have done earlier.
a thorough study has to be conducted on their efficacy and safety, and then only they may be accepted.
If there is any chance to work together, we are ready. We have a methodology for making herbal particles in nano size, and the study can be done at your place, and join the proposal if possible. We can take it forward. Next, I would like to this international bridging heritage, ancient science.
Ancient research and
Advanced technologies. So this will be our on the occasion of our Silver Jubilee. We are going to conduct this in 31st August and 1st September. I am requesting you all, please take it into all your groups, and I request you all, who you are doing like this work, similar work, bridging gap. I would like to please connect the people, and every week, every Saturday, 5 o'clock, this science and technology. So please connect us to everyone, and we would like to start one common dayas. Where can we work together, and how can we hand over this knowledge to next generations also. So thank you. Any more questions?
So Dr. G.B.K. Rao is there. I'm sure he will.
Namaskar ji.
Namaskar.
I am so glad that you have brought so much knowledge and bringing enthusiasm to the people. Dr. Sastry Garu and all are taking such an initiative. Our main focus the all endangered plants, which we have to make it publicity that these are all valuable products can be made, and the people who are growing the plant, they do not know which plant is better for Ayurveda or other valuable products to provide. So we have to take up every year the top 10 plants. We say we should make that top 10 plants which are going to endanger to publicize and see that they are again reproduced.
So you have done commendable job of raising a large number of medicinal plants at Pragati Resorts.
Thank you. That is what Charaka, what he said, there are 55 diseases. For each disease, there are 10 varieties of plant. Each plant can take care of that disease. Out of 55 diseases, 55 into 10, 550 plants. Out of 550, 100 is common. The balance of total 450 varieties, which is going. As per Charaka, whatever he said, for all diseases, 55 types of disease, these 450 varieties of plants will take care. So that is where we have to make it as a commercialized means not for money's sake, but the need of the hour. Instead of allopathy, should bring Ayurveda, Unani, homeopathy. This is our Indian traditional medicine from the plants.
Yes, I also visited Pragati Resorts and heartfelt, very congratulations, doing a great job by Dr. GBK Rao.
Thank you.
And
Thank you.
I also known him, and a wonderful job he is doing. He is one of the, I think, he is the only person doing in India a big resorts with all Charaka Samhita and based on the our Ayurvedic text. He is doing a lot of service and all those things. I
Yeah,
congratulate.
this is our ancient herbs. Trimurti Vanam, Panchavati Vanam, Panchabhuta Vanam, Navagraha Vanam, Nakshatra
Yes,
Vanam,
yes.
Rasi Vanam, Saptarushi Vanam, Ereka Patrika Vanam. All these plants are highly medicinal value. They are
Yes.
connected to the god so that the people will not forget the plants to grow and they don't destroy the those like Tulsi, Brahmi, or Amla, or the type of important plants. So certainly we have to promote that the Vedic, whatever ancient herbs which have been given, like Jamvanta informing to Hanuman to bring that Mudhasanjeevani, Sanakarani, Vishalyakarani, Suvarnakarani, all these plants, the names we have forgotten, and like Somalata, these type of plants we're supposed to again highlight and bring the cultivation more and more.
Very nice, GBK Rao garu. Thank you, and once again,
It's a great pleasure that you are all bringing such type of program.
yes.
So the session today, particularly the Saturday session, evening session, went on extremely well with the presentations given by Professor Reddanna garu and Professor Brindavanam. And here the focus was on the traditional knowledge system and its application in the modern medicine. So several plants which have been enlisted by Dr. Brindavanam and related medicines that have been derived. With this, we are understanding how our ancient, not knowing the importance of the modern medicine, were using this for their health benefits. So like this, sir was mentioning about aspirin, digoxin, morphine, vinblastine, what is that called? Vincristine and artemisinin. So the drug recently, Chinese Nobel laureate, Yu Tu got the Nobel Prize and artemisinin. Like this, several of these drugs have been developed, and only one worry that I'm having, because the people like you all have to take up, that the biodiversity conservation. So that's what sir was mentioning very neatly, future of our drugs. So we may get the food, but we may not get the drugs hereafter, because if we don't maintain our biodiversity well, so we may not get the drugs. The food alone may not make us to survive. Okay. So future drugs depends on the conservation of the biodiversity, sir was mentioning very clearly. So along with this, Professor Reddanna, an eminent scientist who has been doing work on COX and LOX enzymes related to the cancer, and he neatly elaborated the inflammation. What is inflammation? Redness, and again pain and swelling and several other parameters. So apart from that, sir was mentioning a lot of other drugs, and he has also told us about the Branco tea that needs to be used for our health benefits, so that everyone can follow it up, and again you can promote to our friends so that it will be useful to our society. So thanks a lot to you all for introducing us the traditional system along with the modern ways of investigating the traditional system for the human benefit. So as I was mentioning to you, that we can get the food, but we may not get the medicines hereafter. So that's what we have to conserve our, what is it called, the ancient gift heritage. So with this, I thank both the professors for having been sharing their time with us today and making all of us to know about the latest aspects of the Indian tradition.
All. Thank you.
Thank you, Professor Reddanna garu and Dr. Brindavanam garu, and each and every one who participated in the webinar. And please let us take it into the continue meetings every Saturday. So please be with us. Thank you all.